Hi, I'm Yasmine Zeroual, PhD student at Center of Infection and Immunity of Lille. My poster is about use of weighted gene co-expression network analysis (WGCNA) to identify markers of protective respiratory immune defenses during immunotherapy of bacterial pneumonia. The respiratory tract infections and pneumonia represent the third leading cause of death worldwide in infant and elderly. The amoxicillin is known as the first-line treatment used against pneumonia nowadays. That over the years caused the emergence of antimicrobial resistance. The project FAIR I am involved in aims to develop an alternative therapy against this antimicrobial resistance using an adjunct therapy that combines amoxicillin to flagellin. My project is about further understanding the underlying mechanisms of this combination therapy by applying WGCNA pipeline to find markers in blood and in respiratory tissues of protective immunity against pneumonia, all of that in a in vivo murine superinfection model. For this aim, we used a raw expression matrix of 44 lung samples. We filtered the low-quality genes and normalized the dataset using the variant stabilizing transformation VST. Then we applied WGCNA pipeline from the WGCNA-R package with a soft threshold of 8 and a correlation of type B-weight mid-correlation to build a signed network. The module treatment correlation was assessed by Pearson correlation, and the significantly correlated modules to each treatment were selected for the screening phase. Only genes with gene significance higher than the absolute value of 0.2 and a module membership higher than the absolute value of 0.7 were kept. Then, gene set enrichment analyses were conducted on the resulting genes using gene significance as a statistic for some interesting modules. In parallel, genes were intersected with the result of kinetic DEX previously calculated by ISMB software, where each treatment was opposed to a control group of mice only infected with influenza A virus. The resulting gene list consists of genes that are simultaneously highly correlated with each treatment and with a high module membership and also differentially expressed for that specific treatment. These genes were considered as primary candidate biomarkers, and protein-protein interaction network was constructed for these candidate genes. Hub genes were also selected through CytoHub A plugin in Cytoscape. As a result, the heat map of module treatment correlation showed many interesting modules correlated positively and negatively to each treatment, as well as some modules correlated to two treatments simultaneously, but in a different direction, showing the different effect of the treatment on the same set of genes. All modules with p-value less than 0.05 were selected for further analysis. A gene set enrichment analysis was conducted on some interesting modules with a relative large number of genes, as the yellow module in here, that showed a positive correlation with amoxicillin treatment and negative correlation with both amoxicillin and flagellin treatment. We could identify interesting immune-related pathways differentially associated with the two treatments, with apparently a different number of genes involved in the same pathways. That highlights also the different effect of the two treatments. In the end, we constructed the protein-protein interaction networks of the final list of genes selected for each treatment using string database to look at the interconnectedness of the genes and identify the hubs. Many immune-related genes were identified as hubs, and we could see that we had a higher number of genes associated to the new therapy compared to the standalone treatments. Some of these genes are currently being validated by qPCR for more robustness of the results. As a conclusion, our time and treatment resolved analysis of lung transcription by RNA-seq in a in vivo murine model defined potential biomarkers of immunity. Immune pathways are highly enriched with the new combined treatment compared to the reference standalone treatments, showing a potential synergistic effect. The markers are highly correlated to each treatment and differentially expressed at all time points 4, 8, and 12 hours. For the blood dataset, we conducted the same analysis and we obtained also a set of potential biomarkers. A validation of these biomarkers for both lung and blood will be performed by qPCR approach to increase the robustness of the predictive values. And as a perspective, we will be comparing the modules of lung and blood to identify any module conservation or compartmentalization. Also, we want to apply this analysis pipeline in various other animal species to define whether this innovative treatment is specific to the mouse model or conserved through animal species.